that information that has never been available before.
That's fantastic. And so that funding begins now and is staying. It's ongoing for several years.
It's originally a 2-year grant as a pilot, with the idea that if the pilot goes well, then we would work with the state and the CDC and different entities to continue more funding for it. Very cool.
Well, and so I can see we're now being live streamed. So just as a little bit of intro, what we were talking about kind of during the pre-show was how Michigan Tech became a COVID testing facility that opened, as you said, April 9th, 2020, I think,
Yes.
And that was running until we were able to shut it down in December of 2021. And it was basically how people were able to be tested up here in the Upper Peninsula of Michigan. Because before that, as Caryn was relaying, it took 14 days for a test to come back from Detroit, which was just basically not very helpful, as we all know. All right, so welcome everybody. Welcome to Michigan Tech's Husky Bites, and this is season 5. I am going to start sharing screen and just give a few introductory slides. Oh no, opens this slide. I'm going through this again. Sue, I'm having the same problem I had last week, and it's going to be the same link because I didn't fix it 2 weeks ago. Copy link. I'm going to put it in the chat and Sue will share screen. This is what happens when you update your software. I'm like, why did I do that? I've had so many glitches. But yes, this is Husky Bites. We are in season 5, and this evening we're going to be hearing about viruses and how to make a vaccine, how to manufacture a vaccine. And as I said, my name is Janet Callahan. I'm the Dean of the College of Engineering here at Michigan Tech. And so Husky Bites is a free webinar series. This evening's webinar is sponsored by the Department of Chemical Engineering. If you ever want to sponsor a Husky Bites, you just send an email to engineering@mtu.edu, and you can direct your sponsorship to any aspect of the university you want. And as a default, it goes to student scholarships. Next slide. All right, so we had a pretty big ice storm where I don't know exactly what the measured quantity of snow was, or ice, but I don't know if you can see the glazing across this entire meadow. But it was firm enough for the dogs to walk on, and I have a dog that weighs close to 100 pounds, and so it was quite the ice storm. I don't know if you ended up with a glazed over car or anything like that. Luckily, my car is parked in a garage. Next slide. I got a few weather slides. So this is the big icicle that I had until today. Today it got warm enough, and that icicle finally melted, so terrific icicles hanging off of the eaves. And right now, as of this morning, we were at 284 inches. In the season, but it's really, the banks are densifying rapidly and pulling back. This is the time of year where they're pulling back. So it's kind of exciting to see the effect of the sunlight and sublimation. Next slide. All right, and to give you an idea of the height of the snowbanks, this is kind of what the dogs have to do in order to do their business. They have to climb up that very big ramp. I would say it's got to be 5 feet at this point. And you can see how much energy they have. This husky gets energized in the snow. So that is the weather update in Houghton, Michigan. And let's go to the next slide. I do want to let you know what's up next, which is that we're going to be hearing from Sarah Green, who is a professor in the Department of Chemistry. This is going to be kind of telling the tale of some students who with the group from Michigan Tech were selected to join the United Nations Conference of Parties in November of 21, which was held in Glasgow, Scotland. And so we're going to hear directly kind of from the students, and also from Dr. Green, who was the person kind of behind us being involved in that particular event. And the students who will be speaking, one of them is a current PhD student, and the other one is a recent graduate from civil and environmental engineering. All right, next slide. And I think go ahead and stop sharing, Sue. So at this point, what I would like to do, and it is my sincere pleasure, is to introduce faculty member, chemical engineering faculty member, Dr. Caryn Heldt. Caryn is the James and Lorna Mack Endowed Chair of Cellular and Molecular Bioengineering. And so what an endowed chair is when a donor has given a very substantial amount of money to the university, which is called an endowed chair. And then how that helps the faculty member is able to use basically the interest on that endowment in support of their research. And so our foremost faculty hold these positions, and Dr. Caryn Heldt is one of these. She is the director of the Health Research Institute, as well as being the James and Lorna Mack Chair in Bioengineering. A professor, as I said, in the Department of Chemical Engineering. She is also an affiliate professor in biological sciences here at Michigan Tech. She is also an alum of Michigan Tech, having earned her BS in chemistry and chemical engineering from Michigan Tech. And then she went down south to North Carolina State University to earn her doctoral degree. She then spent some time at Rensselaer Polytech before coming here as a faculty member. And I could say a lot more about Caryn, including how she was really instrumental in establishing the COVID testing facility, which basically helped keep our doors open here during the pandemic and helped keep all of us safe up here in the UP, because if we thought we were exposed, we were able to get tested. But I won't go any more into that. I'm going to hand it over to Caryn to introduce our co-host Dylan and to share her presentation.
Well, thank you very much, Janet, for that wonderful introduction. What Dylan and I decided we were going to do is I was going to give you a little bit more about myself, and then I was going to introduce Dylan. I'm originally from Pinconning, Michigan, so a Michigander through and through. And so I was really excited to be able to move up to Michigan Tech as an undergrad. I was recently asked, they're like, so you probably could have gone to University of Michigan, and why did you choose Michigan Tech? And honestly, I was just a rural girl, and I was not ready for the big city of Ann Arbor. Having been here, though, I'm super excited to all that I learned. I think I learned teamwork so much at Michigan Tech. That was a really important part of my undergrad experience. But after I went to Michigan Tech, if you look at the map, I started out working for BASF for 2 years. And so they sent me to Southeast Michigan. They sent me to Texas, went back to Southeast Michigan. In that time, I married my husband, Gary, who I met at Michigan Tech. He was also an undergrad. And then we went to North Carolina. We went to New York. And then we finally came up to Michigan Tech. We did make a big joke when I first moved to Michigan Tech that the next place we needed to move was St. Louis, because we kept making this circle around his parents that were in Southeast Michigan, that were at this 10 to 15 hour range of driving. And then in 2016, I ended up taking a sabbatical in St. Louis with Pfizer. And so that was, I remember sitting on the plane going to my interview going, wait a minute, I said the next place I'm moving to is St. Louis. And I said that in 2010 when I moved to Michigan Tech. So it's quite ironic that happened. But here's my family, my wonderful husband, my 3 children. The goal is for me to be the shortest, which I think we're going to accomplish very soon. They were each, I laugh, their birth certificates look like my resume. I have my North Carolinian, I have my New Yorker, and I have my Yooper. We love to ski. We love just the outdoors in the winter here in Houghton. And this is, we also love geocaching. You would not believe the cool ruins that are out in the woods around here. This was an old train depot that's out in South Range. And so with that, I wanted to then introduce my partner in crime here, Dylan Turpeinen. So Dylan was a student in my lab, And he's currently a scientist, and I think I saw recently promoted to scientist 2, I think, in downstream processing development at National Reliance. So Reliance is a biotechnology company that helps clients develop and scale up vaccine and viral vector production processes. So Dr. Turpeinen previously worked at Thermo Fisher as a quality control scientist, and his PhD work that he'll talk about a little bit did a lot of different things, but he finally settled on aqueous two-phase separations for viral particle purification. And so Dr. Turpeinen completed his BS in chemical engineering at Michigan Technological University in 2016. And then he continued to do his PhD with myself and Dr. Julie King when he got his PhD in 2020. So with that, Dylan, I will let you take it away. I will go to the next slide.
Thank you. So yeah, right after I got my PhD, well, actually right before, right when the pandemic hit, I scooted out of Houghton and came down to Gainesville, Florida, where my wife, Lilu, some of you may recognize her. She's also from the chemical engineering department. We met in grad school. I moved down to Gainesville. She's currently at the University of Florida, and we're both huddled down here in the heat of Florida. Couldn't be happier in the warm weather. So that's us up at Lake Superior. Always have to remember where we came from, the cold water. And then that's us in Singapore. I was lucky enough to go to Singapore over a summer to do research from one of Dr. Heldt's programs. And so she got to visit me over there and do the touristing things for a few days to see all of the awesome things that Singapore has to offer. And then I always forget to point it out, but there's like a built-in picture right here. This is Singapore with a lightning storm in the background. Absolutely loved it there. My family is shown in the middle here. We have 2 fur babies. Her name is Ellie, the dog at the top. We got her, she's a pandemic puppy, so she's super clingy to both of us, but she's a big couch potato. And then Momo, when I was working at Thermo Fisher, she was wandering around outside below a forklift. And so we scooped her up and brought her home and rescued her. And she's everything a feral cat you think she would be. She's feisty. It was one of her rare moments of sitting still. And then for hobbies, what I like to do, I really learned this in high school when I took a woodworking class, but then through undergrad and grad school, never had the time or the space to really do it, but got back into woodworking. So Lilu and I took on this challenge of building ourselves like this little elevated bed. We put the stakes in and we built around it, put some copper caps on the top to pay homage back to Lake Superior and the copper. And our plans for the plants in there are hot peppers. I'm a spice head, so we're going to grow everything from jalapenos up to Carolina Reapers and see what I can handle. And then my last one, showing my super nerdy side. I love PC gaming and I love building PCs. So this is just the internals of one of my old ones. I've recently upgraded it, but just a nerd at heart. I will throw it back to you, Dr. Heldt.
Okay, thanks, Dylan. So since we were going to talk about making vaccines, we're mainly going to talk about the flu vaccine. And so I have the first poll. So I think Sue should be able to pull up the actual, there it is. So the first poll question is, how many flu vaccine doses do you get from an egg? 85% of our influenza vaccines are made in eggs.
So I was looking at this question earlier and I was applying my testing ability to this. And I'm like, wow. So I want to answer 1 because it's such a low number. But I have a feeling that can't possibly be right. So I really don't know what I would answer if I was really having a test on this, Caryn. The majority seems to be for C, which is 5. So the choices are 1, 2, 5, or 10. All right, looks like we've had the majority of the people participate.
Okay, so it looks like we had 44% people say 5 and 38% say 10. The answer is 1.
I didn't Google it.
Actually, in some places, you see it as low as 2 eggs per dose. So I'm going to close this.
Why?
Well, let me tell you a little bit. We'll get into why. So if we need so many eggs to make all of these flu vaccines that everybody's taking around the world, and the eggs all have to be the same age. And so imagine just logistics of having to get all of these eggs to a manufacturing plant. They have to be the exact same age, the same size, all of these things. So there's a different way to make vaccines, and that is what Dylan and I focus on. And so another way to make vaccines is with biomanufacturing. And so biomanufacturing starts with upstream, then you go to downstream, and then you go to formulation. Your upstream is where you take cells, you grow them in these bioreactors, and then if you're going to make a viral vaccine like influenza, you add a virus. You add the virus you want to make, it grows in the cells, and you can use all different types of cells. You can use other animal cells, insect cells, yeast, bacteria, all different types of cells. They'll give you all different types of vaccines, or even how much you can make in each. And then you have this mixture of stuff. You have the vaccine, but then you also have all the pieces of the yeast or all the pieces of the animal cells. Just like in eggs, you've heard that with an egg-based vaccine, if you have an egg allergy, then you can't take it because pieces of that egg are still in your vaccine. And so then you go to downstream. So downstream is where you purify out your viruses and your vaccine or whatever you want to make in your bioreactor. And so now we take this big mixture of stuff where your vaccine may be 0.1% or less of the total stuff in your bioreactor. And now you need to make it 99% of what's in your vial for someone to get a vaccine. And so about 70% of the cost of most biomanufacturing processes are in the downstream. And so that's where my group focuses most of their time. The other thing is that for vaccines, the typical recovery across many different units that will do purification is less than 30%. So there's so much room for optimization and for things to be improved with purification, which is why I love purification and studying it and learning more about it. And then finally, a formulation. Formulation is how you make sure that the vaccine can sit on the shelf. So what do you need to put in that vial? Remember that these are biologics that we're making. And so just like you wouldn't take a steak and put it out on your counter and then eat it 2 days later. That's what so we have to figure out what can we put on that steak so that it could be eaten 2 days later after it sat out in room temperature for 2 days. And so that's really what we're trying to do with formulation. You often add salt, are very common, other stabilizers. You can add sugars, are very common stabilizers. And then often in vaccines, you have adjuvants. Adjuvants are the part of the vaccine that will actually stimulate your immune response. So that's often if you get a fever or anything like that from a vaccine, it's often the adjuvant that causes that, because you need to kind of make sure that your you kind of wake it up and say, hey, there's something going on. Make sure you pay attention to this. And so my group has been working a lot on stabilizers. So we actually received a grant from the National Institutes of Health in February of 2020 to start stabilizing vaccines. And so it was quite ironic when less than a year later, we coming out with these mRNA vaccines that are not very stable at all and have to be put at minus ABC. So we knew before the pandemic that stabilizing vaccines and making sure that they are, our goal is to make them shelf stable so they don't even need refrigeration. And so that's what we're working on. So let me tell you a little bit about upstream. It's not a focus of mine, but I just find some of these pictures absolutely fascinating. This is a bioreactor. And so bioreactors used to be big stainless steel vessels. And now they're starting to use these single-use plastic bags. And you can see that there's an impeller in here and all sorts of different sensors and all sorts of stuff. And they've actually found out that it is actually more environmentally friendly to use a single-use plastic bag than it is to use a stainless steel bioreactor. And it took me a long time to convince myself that this was true. But the stainless steel bioreactors, you have to clean them, you have to sterilize them, and you have to verify that they're clean and sterile before you can use them again. And if you look at a full life cycle analysis, all of that water, all of that steam, all of that waste is actually worse for the environment than taking and making a single-use plastic bag, sterilizing it at the bag manufacturers, and then incinerating it when you're done. And so this is how, when you are taking any antibody treatment, many of your vaccines are made in these big bioreactor bags. So now that I've told you a little bit about a different way to make vaccines than an egg, I looked it up, and an egg is about 1.5 ounces. So if one vaccine dose can be made in an egg, how many flu vaccine doses can you get from a bioreactor that's the same size as an egg? And so the answers could be 10, 100, 1,000, or 10,000 doses.
I don't know. I'm going for the other extreme, 10,000. I have no idea, of course. and nor does our audience. We're getting votes. The majority votes are coming in at 1,000. And then the second closest is at 100, and the third choice is 10,000. So what's the answer?
The answer is 10,000. You're doing great tonight, Janet. So yeah, you can make a very, very different amount of vaccine in these bioreactors. So then, of course, the last question is going to be, why are 85% of our flu vaccines still egg-based? So the answers are: one, A is you get a better immune response from egg-based vaccines. B is manufacturing costs. C, no need to transport egg-based vaccines cold. Or D, we need to keep the chicken farmers in business.
My dog is harassing me. I don't know. I'm not even going to guess on this one.
Not
B.
even going to try,
B?
huh?
B?
So we have just barely each that people are choosing B for manufacturing costs. Okay, so the answer is B, manufacturing costs.
Yes.
That is the main reason. So a cell-based vaccine is 4 to 5 times more expensive than an egg-based vaccine. Having said that, the cell-based vaccines have much better immune response, so they would be a better vaccine. But just we have the capability to do all these eggs. We're used to it. Sometimes change is hard. We would have to build all these new plants. So not only is the vaccine more expensive, but we have to build new plants to be able to manufacture them in a very different way. And so one of the things that our lab really focuses on is how can we reduce those manufacturing costs? And so I'm going to let Dylan tell you a little bit about continuous manufacturing. So that's one of our goals, is to take these batch processes and to continuous to really reduce the manufacturing costs.
Yeah, and exactly what she said is when you switch from this continuous production process from batch production, your facility footprint can go down. You can see that right here on this figure, where on the right side, the gray box area for upstream and downstream suites are much larger than the continuous production suites on the gray boxes on the left. And where this mainly comes from is holding tanks. When you have a batch production, when you go into a unit operation, right before that unit operation, you need to have a tank that is holding what you're going to put into the process. And then through your process, when it comes out, you have to have a tank for where it comes out. Another big vessel is your intermediate buffers. So for your media, you need a lot more media for a batch process bioreactor, and you need a lot more media for your chromatography steps for batch mode production. For chromatography, you have multiple cycles you have to run through for buffer washes to get impurities out when you're actually purifying. And all of those vessels take up space, and you have to have it in a GMP environment, which is super expensive per square foot. And so if you can switch your facility from a batch production facility to continuous for the same product output, your manufacturing costs are going to go way, way down. The problem is actually developing one of those processes and getting a company to buy in and do it and see those savings. And so what we could do is improve the current downstream processing train. And so right now, the current downstream processing train right out of the bioreactor starts with a clarification unit. It will then go through some filters for ultrafiltration and diafiltration. That's UFDF. There are a few chromatography steps. There can be 1, there can be 4. So for 1 and 2, each time you do a chromatography step, you have those buffers, you have a lot of storage space. And then at the end, before you go into your final filtration or your final filling, you're going to fill it one last time and get it into the buffer it needs for the vaccine. And so for a few pictures of what these unit operations look like, and keep in mind, these are smaller scale pilot lab size units. So in the left there, you see a centrifuge. That's a very common unit operation for separating out insoluble impurities from the bioreactor. So you're spinning out things you don't want and collecting the supernatant on top of the liquid, which has your product. And on the right side, it's a little hard to see, but you've got more stainless steel and you've got a filter in between those. And your filter is removing also undissolvable impurities. Now your chromatography, these are also small scale, so they only get larger from here. On the left is an ACTA system, and in this black box that you see, or I guess white box, it's a bunch of valves and pistons and pumps that will pump your different liquids and buffers through the column and then into different outlets. So it's a very complex system in there, and it only gets more complex when you scale it up. On the right side is a typical pilot size plant for a chromatography column. And that column right there, what you're going to be looking at is going to represent a large cost of your downstream process. That resin in there, I don't know what resin it is, but if you're looking to buy 10 liters of resin, you're going to be spending $250,000 just for one column, which is likely single use, and then you get rid of it. So that's where the big costs come from. And through all of those steps, it's less than 30% recovery through them. As you can imagine, each step has its own challenges for yield loss and recovery. And typically in industry, and I see this through the processes that I run, yeah, 30% is like very good. Like we hit them, we got it at 30%. So we got to do better than that. And so what I worked on in grad school was developing a different downstream processing train focused on aqueous two-phase systems. That's a fancy word for liquid-liquid extraction, which is a fancy word for oil and water don't mix. So if you put something into a system of oil and water and something is more soluble in the water or oil and you let those phases separate out, you achieved a separation where you can collect one of the phases that has your interested product. For ATPS, you can do one stage and then a second stage of the same process, which is called back extraction, to get it into a more favorable phase. And then just do a simple little filtration to get it into a final filtration, final formulation buffer for filling your vials for vaccines. And with this, if you can, double it just to 60% recovery, you're producing twice as much with half as many steps. So that's what the goal was to improve the downstream train to get more out and have the manufacturing costs less. And what this looked like is you start again in the upstream. You can produce your virus in a bioreactor. And at the end of the bioreactor, you have all of your junk from upstream and your product of interest, which would be a virus. You can concentrate it down a little bit to make it easier for your purification train. And this would just be removing bulk solution to get it out of the large size of a bioreactor. And the ATPS here was the main technology and the main system of what we were focusing on. And the eventual goal and the hope is that this ATPS system, running at 1 and 2 stages of it, can basically replace all of those chromatography steps. Because in essence, those chromatography steps are just trying to separate out soluble impurities from your product of interest. And this is the same thing that aqueous two-phase systems can do. And what we were looking at was using a polyethylene glycol, a PEG, which is a super viscous polymer. Think like honey, but like cold honey. So it's really hard to flow and super viscous. And then we have citrate, which is a salt. And when you mix PEG and citrate in certain ratios with a virus that you want to separate out from other impurities, you get a mixture. And after they settle out, you can collect PEG and citrate separately. And what we can do is monitor where the virus partitions or favors. And what we found out is that it favors the PEG-rich phase, which you can then take on to certain other unit operations to remove the PEG and then to put it into a final formulation buffer. And so what that looks like in a non-schematic way, in actual lab application of what you can do, this was the benchtop picture that I took in the lab. I set this up, and on the left side, you see the feed pump. It's a peristaltic pump with tubing that pumps in that PEG, citrate, and virus mixture. The helical mixer in the middle is a static mixer, which allows the phases to have proper contact and have diffusion of your particles where they need to be. So your contaminants in the citrate waste phase and your viral product of interest in the PEG-rich phase. The settler in the middle there with the purple cap, you can pull out the top with a pump and pull out the bottom with a pump, and that achieves your separation. And this is what that looks like. We dyed the PEG-rich phase orange, and then the citrate-rich phase would just remain colorless. And so right in that settler, you can see these settle very quickly, and you can collect right off the top with pure PEG, and then out the bottom with pure citrate. And so for just some bonus downstream pictures of what these large facilities look like in real time, on the left here, this is one of the clean in place and steam in place setups that would be needed for a traditional stainless steel reactor or equipment, where there are large tanks of sodium hydroxide that you use for cleaning your solutions, and then you collect it in a different one, and then you have to steam all of your stainless steel. I will say that I have had the pleasure of not running one of these because Resilience is a single-use only site, so we go with the plastics and throw those out when we're done. And then on the right here, you can see a chromatography column, which is on the 3 little wheels with the resin in the middle again, the super expensive resin, hooked up to the chromatography unit, which has its valves and pumps exposed there that you would actually be there typing into the computer, setting the flow paths and making sure that you are flowing in the correct direction. And so that's what I'm doing almost every week, is running these chromatography steps.
Well, thank you, Dylan. And so that's a lot of what we're working on with our downstream. And then I also wanted to kind of mention a little bit of what we're doing with formulation. So as I mentioned before, that a final vaccine formulation, you have an antigen, which can be your virus, your RNA, whatever is causing the immune response you want. You then have an adjuvant. So the adjuvant then just boosts your immune response, so it's stronger. Most adjuvants are based in aluminum. And then you have your excipients. So your excipients are your preservatives, your buffers, and then stabilizers. And so your stabilizers are often sugars, sucrose, trehalose, can be glycine, which is an amino acid, or even sometimes it's serum albumin. This is actually the main protein in your blood plasma. And so if you give plasma, you're really giving a lot of albumin. And so this is our full vaccine formulation. And the reason that we are really interested in stopping the vaccine cold chain. And so what the cold chain is that it's actually a requirement for the vaccine to stay at a certain temperature throughout its transportation cycle. And so every step of the way from the manufacturer, and then you have land transport to a storage site, and then air transport to another storage site, then land transport to your clinic. Even at the clinic, they have sensors inside the boxes the entire time, and they have to stay cold. So depending on the vaccine, the Pfizer mRNA COVID vaccine needed to stay below minus 76 degrees Celsius. Often they need to stay between 4 and 8 degrees Celsius, which is the temperature of your refrigerator. So you can imagine that one, this is really expensive to maintain all of these temperatures at so cold. And also we lose about 50% of all vaccines due to deviations in the cold chain. And this includes vaccines in the United States. So very often, even vaccines transported within the United States have to be thrown out because there was a deviation in the cold chain. So I've been working with a fellow chemical engineer at the University of Massachusetts Amherst, and she works on these polymers called coacervates. And so basically what a coacervate is it's a dense liquid phase. So as you heard from Dylan, we love liquid extraction and liquid phases. And so this is another application of that. Now, when we take a virus and we add this positively charged polylysine to our virus, and then we add a negatively charged polyglutamic acid, the negative charge of the glutamic acid and the positive charge of the lysine combine and actually make these liquid droplets on the bottom of a well or a plate. And so it looks like oil droplets basically on the bottom of a if you had water there, except they're more dense, so they sit at the bottom. And we have found that the viruses that are captured in these coacervates can withstand temperatures much more than can a virus that is not in one. And so we are still working on understanding why that is. Our guess is that this mimics the natural environment of your cells. Your cells are very, very crowded. And so it's well known that a crowded environment for proteins and for vaccines will often keep them stabilized. And so that's one of our hypotheses, but we're working on several to try to understand why this happens so that we can improve it and make it even better. So that's really kind of my overview of some of the things that we're interested in vaccine manufacturing. Starting with a little bit with upstream, and then talking about the downstream and our focus on continuous manufacturing. As Dylan mentioned a lot about chromatography, there's a lot of people that are working on making chromatography continuous. It is continuous to load a column, but it is not continuous to elute off the column. And so you can have a continuous everything before your chromatography column, but everything after that is not. And so that's why we have been focusing on aqueous two-phase extraction, because we wanted to find a way that is completely continuous coming in and going out. And then we also are interested in formulation and the way that we can make vaccines more temperature stable. And ideally, we would love to have them shelf stable. And so with that, I want to definitely take the time to thank all of my group members, both my current group and my past group, all of our collaborators from Michigan Tech and from many other places, the great students that I get to work with, and of course, thank our funding sources that allow us to do all this fun work. And so that's the end of our presentation. So we're, maybe I'll stop sharing my screen, and then we can start thinking about answering some questions.
Thank you so much. And so folks, you can enter a question by moving your cursor down to the bottom of the Zoom area, and you'll see a tab or a button that's Q&A. And you can ask a question, or if you just have a comment you'd like to make personally to one of us, what we'll start doing is answering some of these questions. And so Dylan, if you pick an answer to question, you click on answer live, and then once the person finishes answering the question, you'll indicate that it's done with asking questions. So we've already got 3 questions up there.
All right, let's see how this goes, because I've never used this feature in Zoom. So let's see.
Well, I definitely want to answer the question from Roseanne, because she was one of my students. So great to see you. And yeah,
So wait,
her question.
So yeah, because the audience can't see the questions.
Right, I was just gonna tell her what the question is. So yeah, her question is, how much does the plastic bag leach into each batch? And she's asking because plastic leaches chemicals under heat, like microwaving. And so leachables is actually a huge issue. And so all of the bags are tested for their leachables. So it's quite known that a certain manufacturer has a certain kind of profile of leachables that are going to come out of their bags. And what then happens is those they have to be tested for at the end. So when you have a final formulation, you know that if you have bag X, you have to be testing for A, B, and C out of your leachables. And so you test for that at the end. Typically, they get removed in the downstream process without really trying. But Dylan, do you have any experience of trying to remove leachables?
No, because like you said, yeah, they'll just come out in one of your processes, since there are so many steps already incorporated into the train. But I do want to say that, yeah, the leachables are tracked. When like a procurement team gets these bags into a facility, they go through C of A's and C of T's, which are certificates of analysis and testing, to make sure these leachables are within tolerable limits to be used in a good manufacturing process facility. And then, yeah, the quality control team has assays at the end of the process to test for these leachables.
All right, and now I'm going to inject my 2 cents. So never microwave plastic things. Open up the plastic thing, put it in a glass bowl, put a plate on top of it, put some water in it, put it in the microwave. You know, it doesn't take very long. Don't microwave things in plastic. It's not good for you.
The highest temperature that these bags will see is at the bioreactor stage, which is around 37 C. So it doesn't get too hot, but yeah, you can still get some leachables at that temperature.
Which is body temperature.
Yes. Okay, there is this question that I don't think I can really answer. Is there any ongoing research at Michigan Tech to explore viable plastic alternatives that are compostable that may be used in your continuous vaccine production system?
So not that I'm aware of, but the biggest problem is that the bags are then biohazards. So even if they were compostable, they have to be decontaminated, which is why incineration is the chosen way to get rid of them, because then it both decontaminates them and is the way to get rid of them.
Yep.
So I can take the one from Marie on have you considered using simulated moving bed technology? So SMB is actually what we were kind of referring to as what is continuous chromatography at this point in time. So simulated moving bed technology is basically takes several chromatography columns. You load one with a stream, and when that one's loaded, you start loading the second one. And then while you're loading the second and third one, you can wash your first one and elute it off. And so by the time you get done filling your last one, the first one is all regenerated and ready to be used again. And so that's what we were referring to, that chromatography can be continuous into the chromatography step. But because you have to wash and then elute, it's not continuous coming off of the chromatography step. And so that's the biggest challenge there.
I'm going to guess that Mike asked that question.
I will also say from an industry side, we do have an SMB unit in the PD lab that I work in. I don't personally use it, but I know the scientists that do have to work on it do not have a good time. It's already difficult enough to get one column operating smoothly with pressure profiles and flow rates. That thing is alarming constantly when there are 8 columns going that you have to try to get going at the same time.
So I'm definitely going to take this this comment from Glenn Lawrence. Hey, Glenn, saying that single use versus stainless steel reactors, cost effectiveness is dependent on capacity. And that is absolutely correct. The largest single-use bag that you can get is about 2,000 liters. And stainless steel reactors can be 10,000 liters, can be larger. So for example, Genentech is a leader in antibody production, and they make a lot of antibodies. And so they are working in 10, I think I even heard them once say even 20,000 liter bioreactors. And so, yes, Glenn, you're absolutely correct that when you are in very, very large capacity, that the stainless steel is better. But if you want to be a smaller facility, like Dylan is doing, that has a lot of different products going through it, and you want to be flexible, then that's when the single use is much better.
I think this question is pretty interesting. As costly as it is to research these processes, are the results shared across the competition? The answer from an industry side is, of course, no. It's inside knowledge. It's not shared with anybody except for the client and the person you're working with. And so I say client and the person you're working with is because Resilient is a contract drug manufacturing organization, a CDMO, where we have clients come in with their technology and we produce it at a larger scale. So it's only shared between us, and it's not shared between anybody else. Now for the research side, with publications, if groups decide to go the route of publishing their results and sharing that knowledge, the aqueous two-phase system that I talked about during research or my research talk, that's published. You can find that research out there. It's really up to the individual if they want to share it or not.
So there's also a question on if there's common processes between vaccine purification and metallurgic extraction. I like that a lot. If you're familiar with the chemical engineering department, we have several people that actually work in mining engineering and in metallurgical extraction. And we talk all the time because, as you can see, Dylan and I do a lot of extraction processes ourselves. And I talk to them a lot too, because the production of iron about 50, 60 years ago went through the same transition of going from batch to continuous processing. And so as the bio area starts making that type of transition is when we start to think like, well, what can we learn from other places that have done this and try to make these batch processes continuous?
So there was an earlier question here of, can you go through again how Michigan Tech ended up being getting certified to do COVID testing? I don't know if you want to answer it briefly or just refer back to the recording. Up to you.
Janet, do you want me to go over that again?
I think taking a minute to go through it. Sorry, I'm muting because my dog is like moaning at me because she's so bored. So I'm gonna stay muted.
Yeah. So what happened with the COVID testing lab was right after shutdown. It was not you couldn't get a COVID test in the Upper Peninsula of Michigan. Detroit was one of the first hot spots, and the only place for tests to be run was at the state labs in Lansing. And so they weren't prioritizing samples from the UP because there wasn't any known contacts. So only if you had a known contact could you actually get a COVID test at that time. And so I was approached, as were many other faculty on campus, to try to understand if we could do COVID tested safely on campus, and two, if we had the equipment to be able to do it. And I was the expert on the safety because we grow biosafety level 2, which are organisms or viruses that can affect humans, and but are typically do not cause severe disease. That's biosafety level 2. 3 causes severe disease, and 4 is like Ebola. And so we found out that the testing was biosafety level 2, which is something we can do at Michigan Tech. We cannot do biosafety level 3, which is research with SARS-CoV-2. And we found out that we had 2 real-time PCR machines, which is how the PCR test is run. And what was interesting is we called around. We had the only real-time PCR machines in the Upper Peninsula. We asked Northern, we asked Lake State, and they didn't have them. And after that, it took us 3 weeks from our first phone call with several, many days. We had 2 conference calls a day for updates, asking questions. But it was just amazing the contacts we had between the people in forestry and biology that understood how to do real-time PCR, the medical laboratory sciences program that knew how to intake samples, David Dixon in research integrity who had who knew people at the state labs that were helping us and would give us panels to do original testing on. IT was amazing. And really helped us out, gave us computers, and we burned through like 3 fax machines. we kept getting more fax machines. And so it was really just a whole university effort of we wanted to get this done, and we worked really hard until we accomplished it. And the first few months, we were only receiving samples from the community. We started in April of 2020, and not until the students came back in September of 2020 did we even start running Michigan Tech samples. So we very quickly just became a resource for the community to be able to get tests run. And we were, in most cases, getting results out in 24 to 48 hours, which was just amazing at the early onset of the pandemic.
You kept us open, you guys.
Okay.
Oh gosh. All right. So this is a fun question from Kent. Hello. When I get my annual flu shot that is a 4-strain version, does it take 4 eggs?
I skipped that one because I honestly don't know the answer. I know everyone says that 1 dose is an egg. And I was thinking about that one. you do need 4 eggs to be able, because each strain is in a different egg. And what I don't know is though if that totals 4 eggs or if it totals a quarter of an egg for each
So
strain.
why, I mean, we don't inject the volume of a whole egg in a flu vaccine. Why can't it be more than 1 dose?
It's just how much virus can be grown in the egg. So one of the reasons that the egg vaccines are only about 60 to 70% effective usually is because we've taken the virus that infects people and have changed it so that it will grow efficiently in an egg. And that changes it enough that some years it elicits the immune response we need, and some years it doesn't elicit as strong of an immune response against what's actually circulating. But that's just how much virus actually grows in the egg. And
Okay.
so
it's concentrated from the volume of an egg into
Yes.
a tiny amount. Okay.
it is. Yeah.
That was
During the
bothering me.
purification, it's concentrated.
All right, this question should be quick. Are most flu vaccines administered in the United States made in the United States? Do you know that off the top of your head, Dr. Heldt?
I don't know that one off the top of my head either. I know a lot of it is made in Europe. So I'm not 100% sure. Do you know, Dylan?
I don't, but I if we say most, it's going to be Europe and United States for sure for dominating global.
And perhaps there's been a shift, in the recent climate of, just being a little more ready on our shores. There may be a shift to increasing, the production in the US.
Well, the challenge with that, though, is that these are not very easy plants to build, and they take a long time. They take a lot of money. And somebody had also mentioned that the FDA process and how rigorous it takes to test batches, and the FDA will allow you to actually use this plant is such a rigorous process that you're not going to just have them pop up overnight. It's a multi-year process. Dylan, do you want to make any of the comments about working with the FDA?
Yeah, I mean, the overall is just, it takes so long. It's just the paperwork, the things that you have to check. You have to be rigorous because, again, at the end of the day, these products are going into people. So there's no shortcuts taken in any of these processes. It's combed through like crazy. We had some FDA ex auditors come through the resilience site, and they were there for a week and they found hundreds of things. So if it takes, if they take years to look at things, it just takes so long to get through everything. But I will say that the reason it takes so long is to get it safe. That's the goal. Is we're not trying to rush through any of this. The best thing to do is to slow down and make sure all of these steps are taken to make it a safe product.
And there's a big thank you from a couple of people for the leadership in helping get the testing facility up. And I know, Caryn, you're going to immediately say it was a community effort, but this is some thanks to you personally. So,
well, thank you.
Yeah.
I see several of my students are asking questions. I see Jake is asking about a very specific chromatography resins as being flow through. Have you ever used any of them, Dylan?
I just placed orders for some, not that one. I can't tell you which one. I have to be so careful with what I say because everything is so hush hush at a CDMO. But yes, we use them in industry for flow through capabilities. They're great resins.
So that's the best way to actually do your chromatography in truly in continuous, is to do what's called flow through chromatography. And what that means is that the product does not bind at all. And so the product that you want flows right through, and all your contaminants stick to the chromatography resin. And that is a truly continuous way of running chromatography. What we were talking about before is that typically chromatography is your product sticks to the chromatography resin, and then you wash away other things, and then you change the conditions of the liquid so that only what you want comes off the resin. And that's what is not continuous.
And the challenge with that, with the flow through columns, is if your resin isn't able to be regenerated in a way that you can't get all of what you removed out of that column, it's single use. And again, those are not cheap.
We're running up on the hour. Maybe do you want to just take 2 more questions and then just some closing remarks? Well, actually, I'm going to ask a question. That would be one of the 2 questions. And this is a question for Caryn. So we might have some students out there who are thinking about what they want to major in. And I noticed you majored in both chemistry and chemical engineering. You know, and I was close to that boundary too. And I ended up majoring in chemical engineering, not both. Do you have any comments about just being not quite sure about what you want to major in and why you chose 2?
I honestly, I chose 2 because I had a bunch of credits I brought in from high school. And so I had enough credits for 2 bachelor's degrees. and the one that was closest, or I needed the least number of required more classes, was chemistry with a chemical engineering undergrad. Having said that, though, what having those 2 very different undergraduate degrees really taught me was the difference between engineering and the sciences. With the sciences, it is I felt like it was the learning was very different. You needed to understand and learn all of these processes and the names and these chemical reactions and the way things worked, where the engineering was much more math focused. You needed to be able to understand these equations and how they applied, and using your math skills much more to answer questions. Versus the sciences, which I felt were much more fact-based compared to math-based. While it's not the easiest to figure out what to do as an incoming freshman, you really choose what's interesting to you, realizing that, I'm going to put in a plug for chemical engineering because, of course, that's near to my heart, is that with chemical engineering, there's so many different things you can do. And that's what really sold me on it. You can be an environmental engineer with a chemical engineering degree. We do bioprocessing. And so, we're able to work with the pharmaceutical industry, and we were talking about metallurgy, processing. There's just so many things that you can do with a chemical engineering degree. It is so versatile. And so if you have any interest in chemistry, biology, and math, like chemical engineering is a great place to think about what you want to do.
All right, one question that Dylan picks out, and then just some closing remarks.
Okay, I think we'll go with this one. The very long-term goal of industry is to develop continuous processes for pharmaceutical production, but the barriers are very high. FDA wants to be able to test batches, and the concept of a batch and continuous processing are in direct opposition. Yeah, so this is a question that we also talked about and debated when I was in grad school. And it comes down to, yeah, a batch is super easy to define. What you put in at the end of the process, what you get out, that's your batch. For continuous, you can just shift your thinking into saying, if you want to define a batch, and if the FDA really requires a definition of a batch, whatever you get out of your process in a 10-minute window, in a 1-hour window, that's batch number 1. Then in the next hour, batch number 2, and then number 3. And you can do the same exact quality control assays and tests on those batches and just define them in a different way. But I do know, now being in industry and with these continuous talks of, they want to implement this technology, the FDA is very willing to open their arms and say, please convince us that continuous is the way to go, and just prove it with data like any other process. And so that's where we're going. We need to prove to them that we can do it and that it is worth it.
All right, and so closing remarks from me. Thank you, Dylan. Thank you, Caryn, so much for your time this evening. I learned a lot. It made me proud to be a chemical engineer. Dylan, I'm just going to give it to you first for just some closing remarks, and then Caryn, you can close it out.
All right, thank you. Yeah, I mean, my closing remarks, I'm going to plug resilience. I really love resilience. When they took over Ology, the message that they sent out, and they have want to hit the ground running and develop technologies to really tackle the next world problem with our feet on the ground, really made me interested in staying at resilience and really putting my best foot forward. So with that, resilience continues to buy sites and continues to hire people. So if there's anybody out there looking for career opportunities or internships, just check out the resilience careers page. We are always looking for talented, driven people.
Caryn.
Thanks, Dylan. And I want to thank Dylan for taking the time to talk with us today. Thank Janet for her invitation. And I would say my closing remarks are that chemical engineers can do almost anything. My mom and my sister are nurses, and so I always was interested in the medical field, but I wasn't interested in being a doctor or nurse. And so I have really enjoyed working with Michigan Tech students and bringing bioprocessing and showing them how you can still be in the medical field and the pharmaceutical field as an engineer, and have really enjoyed watching students succeed and go into pharmaceutical sciences, into different companies. And so if that's something that you're interested in doing, I would love to talk to you, and would love to have more people at Michigan Tech who are interested in pharmaceutical engineering.
All right, thank you, everyone. And reminder, next week we've got Dr. Sarah Green from the Department of Chemistry, who's going to be talking about the trip that Michigan Tech students took to Glasgow, Scotland, as agents of change. And so we look forward to seeing you next week. Thank you, everyone.
Thanks, Janet.